So the format, each researcher will have 7 minutes to give their pitch, and then 5 minutes for questions from the funders panel. The funders are not going to be committing at this time to whether or not they're going to fund these projects. This is kind of a first step, making the connection so that we can set the way for future collaboration. All right, and without further ado, our first pitch is Regina Monaco.
Thank you. Hello? Oh, it works. Okay.
Hi, Regina.
Hello, whoever says hi. Hi, everybody. So I'm really excited to be here, and I'm glad to have this opportunity to talk a little bit about some research that I'm interested in doing. Oh, and this works. Let's see. Okay. So I would like to talk about memory retention post-cryopreservation, and specifically what I'll talk about today is preservation of training in cryopreserved mammalian neural systems. So I hope you'll agree with me. I think that preservation of memories is essential for a successful cryopreservation procedure. I know people have different opinions, but myself, I don't want to be brought back without my memories. So I want to answer the question, after cryopreservation, do our previously encoded neural information, does it persist? That's our memories. Do they persist, and are they accessible? So I want to establish that neural tissue is known to survive cryopreservation. There's been decades of meticulous research on this. If anyone wants to know more, anyone here, here's some references that I think are really good. I think that if you read these 4 references, you'll be up to date. But basically, there's not a question that the tissue survives. The question is, do the memories in the tissue survive? So let's discuss the nematode in the room. This is a C. elegans. She's fancy. She curls her tail for us. So C. elegans, a little worm that could remember. In 2015, Vita Moore and Veronica, and I think Natasha's here, or she was here earlier. So they did some fantastic work. I think it was groundbreaking work. They have demonstrated that learned memories in the invertebrate C. elegans survives cryopreservation. So the groundwork for this idea has been laid. I know that C. elegans is like, this big, but it has a surprisingly complex neural system. Even though it has 302 neurons, they're connected in a very dense way, and it uses something called long-term potentiation, which is how neurons encode memories to encode information about its environment, such as places to avoid or finding food. So it navigates its environment, it's dynamic, it learns, it's an important animal. And while, now I'm gonna contradict what I just said, while surprisingly complex and highly functional, this organism is far from being mammalian. I don't think anyone in this room would be like, we're done. We've showed it in C. elegans. I don't need to know. You don't need to test my brain. No, we're not completely convinced. We just have this amazingly evocative work that's already been done. To continue, though, we now need to study the persistence of memory post-cryopreservation in more complex mammalian neural systems. That's going to tell us more about what we need to know. This is just a quick picture. I'm not going to discuss it, but this is the connectome of C. elegans. You can see it's not simple, right? And it remembered post-cryopreservation. So training mammalian neural cultures requires a little bit of fancy footwork. So teaching or training a neural culture on a slide is a puzzling task. So I have a little proxy here. This is a Snapple cap. So just imagine that a neural culture is growing in this Snapple cap, right? It's the neural culture. I want to train it to do something. Okay, jump, right? It does nothing because it can't hear me, right? There's no way to really communicate with a neural culture, especially if it's not really there. Sorry. So the early idea that I had, I've been batting this idea around since 2016. And my original idea was to induce learning using chemical induction of LTP. Remember, LTP, long-term potentiation, is the biochemical process that lays down memories. I had the idea that if you did it with a neural culture, you could induce some sort of patterns or something that would then persist, and you could measure it after cryopreservation. So at this time, I opened discussions with people at Alcor, and Hugh Hickson was interested in this idea, and he introduced me to Greg Fay and others at 21st Century Medicine, and we've been discussing this ever since, and I have to thank them for a lot of encouragement and a lot of good ideas along the way. So the problem with the long-term potentiation by chemical induction was that while it's easy to induce it, the patterns were not persistent. It was not a good idea. No matter how it was played with, it did not induce something that you could test in a way that you would want to. So this method does not encode the system with persistent, meaningful, or testable information. So okay, we want to train neural cultures to do something so that we can then cryopreserve them and test them, right? How do you do that? How do you do that? So it's not the easiest thing. While I'm thinking about it, a new technique emerged in the literature. The bioengineering labs, they have nothing to do with cryonics, of these particular researchers, Potter, DeMars, and then Friston and Kagan, published something extraordinary. They designed a system to train neural cultures. Thank you. This is perfect. I was very excited about this. To play Pong, by the way, I don't really care what it does. If it has a skill, if it learns to do something, I am going to want to cryopreserve it and see if it can still play its game of Pong. So this is a gift. Lest you think I'm kidding, this is just a quick snapshot of the first page of one of their articles. In vitro, neurons learn and exhibit sentience, okay, when embodied in a simulated game world. This is good. So they do this by using the technology of multi-electrode arrays, and that's how the researchers were able to open communications with neural cultures. So I'm just going to show a quick picture. Oh, and that got all mangled. But basically, this is a multi-electrode array, and you can see now why the Snapple cap was sort of a cute idea. So in that ring, that's where the neural tissue would be grown, and it's sitting on top of an array of many electrodes, hence the name multi-electrode array. And these electrodes, these pads, are in communication with a computer. So this goes into a piece of equipment that then has connections to a computer. And the tips of the electrodes are coming through the bottom of the slide like this, for this kind of slide. So you would have, imagine holes in this. So you would have the ability to have the computer send communication signals through the electrodes to the neural culture. And I will tell you that these labs that I just mentioned found a way to train this neural culture to play Pong or fly a plane, which is a different bunch of papers. So I'm going to skip this because of time constraints. We're going to go to a video, which should start moving. Hello, video. Is there any way to click it? Okay. Okay. So this is going to move fast. Ignore this. This is the game. This is an actual game from the Friston lab. It's going to come back. This is their multi-electrode array that they use, and you can see it's in the piece of equipment where it's communicating with the computer. Basically, back to the game, the culture, there's a single culture, and it's controlling the movement of this paddle to intercept the trajectory of that ball. Now, I get that this is not something you'd buy a ticket to watch at the US Open or anything, right? It's a really big paddle, and the ball is like padoodling along really slow. But yeah, I heard somebody say amazing. I think so too. Now, the interesting thing and the important thing is that the untrained neural cultures miss the ball. I'm sorry, they intercept the ball about 28% of the time. That's just random because this covers about a little less than a third of that side. So randomly, that's about the score it's going to get. But after they're trained, they intercept the ball over 90% of the time. The neural culture has actually learned something demonstrable. Now we have a system, we can freeze this, cryopreserve it, bring it back, can it still play Pong, and what's its score? So this is a perfect system. As if there weren't enough good things happening and that now I have this ability to test these systems, Greg contacted me about a month ago to let me know that 21st Century Medicine has just designed some new equipment which allows the application of existing cryopreservation protocols that are already in use for organs and whole brains to neural culture slides. This is fabulous. So what it does is it's new automated equipment that allows continuous temperature monitoring and gradient application of cryopreservants to these neural culture slides, which means that we can now closely replicate already successful cryopreservation methods in these slides. This is an enormous addition to these experiments. This is fabulous. So now you can see the pieces have come together. now is the time. So this work, if it's funded, would be done in collaboration with 21st Century Medicine. Phase 1 is what I'm talking about today. I would like to look at rat hippocampal neural cultures to start, following the protocol of Kagan to train it to play Pong. After it's trained, I would measure how it plays the game. I would take its parameters and scores, and I would define them, measure them, and record them. And then these will be cryopreserved using, according to protocols already known to be successful for neural tissue, and using Greg's new gradient apparatus. Comparison of game scores before and after cryopreservation yields the degree of memory preservation. Boom, this is good. It's very straightforward. So my final thought that I'd like to leave you guys with, this is from a discussion Mike Darwin had on cryopause in 2011, and it was a discussion of studying the preservation of memory post-cryopreservation. Under what conditions is long-term memory and personality likely to survive or not survive cryopreservation? It is astonishing that cryonics organizations have not conducted in-house or extramural studies of this kind on brain tissue subjected to the cryopreservation techniques currently in use. So now all the pieces are here. I would love to do this research, and I hope that you're all as excited about it as I am. And I would like to quickly acknowledge and thank the gang at 21st Century Medicine for collaboration, encouragement, and so many productive conversations. And that would be Greg Fay, Ralph Spindler, who's also here, and Brian Woke, and Hugh Hickson at Alcor, also for encouragement and good discussions. And thank you for your attention.
All right. Okay, we are going to have 3 minutes for questions.
I take it you're using brain organoids?
I would like to use organoids, so I'm going to start with a two-dimensional cell culture that would grow on the bottom of this, of a cell tissue culture plate like this. But the phase 2, I would like to go to neural organoids, and I already have several in mind.
Neural organoids, do you know what the costs are?
The cost?
Yeah.
I don't.
Okay.
Actually,
all right.
yeah.
Thank you then.
Yeah.
How much and how much time do you need?
That's an interesting question. I would say we could get some results in a year, but I'm thinking more along 2 years. How much? Oh, like $503,000.
Sorry, we can only do 500,000. Last 3 you'll have to do somewhere else. Yeah, first of all, super cool project. I think this is one of those ones that can really open people's minds to any of this really being possible. I think some of these experiments are yeah, so this is super cool. I guess if, at CryoDAO we want to, de-risk these projects as much as possible. If a project were to fail, and it's fine if they do sometimes, we'd like it to not fail on something that is not cryopreservation. So I'm curious, how established is this training method that you're using, and would it be possible to even have any assistance from the lab or labs that actually develop that training method for this?
Okay, so first I'll answer it kind of backwards. So I've already been in touch with Carl Friston and Brett Kagan, and there is some possible interest of collaborating with them and getting some training there. But about the idea of failure, I think that's interesting because I believe, and maybe I'm optimistic, we've already proven that cryopreservation, I know in a very simple system, but the brain still retains some memory. There's a lot of evidence in other sort of circumstances in the animal world, and I know they're not completely cryopreserved, but there's a lot of evidence that cooling does not erase memory, even hypothermic cooling, to minus 20 degrees. There's been experiments like that. So I think if this fails, it would be the technique that fails, and that would mean let's know that now so that we can continue to work and find out how to do it so it doesn't fail. Yeah.
All right, that's all we have time for. Thank you so much.
Okay, thank you.
Next up, we have Ramon Risco.
Okay. Thank you very much. It's for me a pleasure. It's for me a pleasure being here. Let me.
He's going back. You're going around.
Okay. Let me start this talk by asking the same question as my friend Greg Fahy did some years ago. Who in this room would like to take a dip in the... Sorry?
Put it up to your mouth.
All right. Who in this room would like to take a dip in the fountain of eternal youth? Probably everybody would like. The thing is that today, this is a reachable reality. However, it will need some time. That's why we need cryopreservation. The problem with the cryopreservation is the presence of ice. So there are two strategies to avoid it. One is to put a cryoprotectant, a molecule that you put in between the two water molecules in order to avoid them to stick together. And the second thing is just to do a very fast cooling, and more importantly, to do a very fast warming. Historically, electromagnetic radiation has been the election for this quick rewarming. Now we have three variants. One is the historic microwaves, in more recent times nanowarming, and very recently the electric heating. All of them are wonderful technologies with very promising results. They share this capability of penetration, of heating the inner parts. That is what you need for having a quick rewarming. However, they also share some drawbacks. One is the problem of the thermal runaways. The second thing is that they cannot be monitored with typical MRI of CT because you have to put metallic parts inside the machines. In the case of nanoparticles, you have had to put the nanoparticles before the cooling process. So we know that now there are hundreds of bodies waiting for a solution without these nanoparticles. So welcome to the future. I think that the future is ultrasound.
How is commonly noticed?
Sorry? Oh, so can you switch off the sound?
Oh, okay. I think that's it. Continue.
So what I was saying is that probably the solution is here. With ultrasound, you have thousands of transducers. Each one can direct the sound and the heat to a very concrete point. The thing is that each transducer can be steered, can be directly, and put the energy exactly in the position that you want. And it can be done with the precision of millimeter and controlled by magnetic resonance image. That is the key point. So in this case, for example, a patient with essential tremor get recovered in question of minutes with this ultrasound technology. The applications today are hundreds. Every year appear new applications of this high-intensity focused ultrasound guided by magnetic resonance. You see here all of them, or plenty of them. So something like 20 years ago, I started thinking on that possibility, and it was not till this meeting in Madrid, in the society, where I present the result that at least for the ovary, you can have in a simulation very, very uniform rewarming with a set of only 27 transducers and with a very, very quick warming rate. This so tight lines means that the rewarming is very, very fast. So after this simulation that were published in this couple of journals, Cryobiology with an abstract and the Journal of the Society for Electronics, I decided to build the setup. This is the first setup. And then I look at the performance. This is the cooling rate, till -80, and you see the warming rate that is practically sudden. It is instantaneous by the application of this ultrasound technology. So I decided to test it in 3 different biological systems. The first one was aortic rings. These aortic rings, after the treatment with ultrasounds, contract as they should, contrary to the regular cryopreservation in banks, that they are dead. These are not simple tubes, they are alive structure. Also, the inner part are intact, and we wanted to see even deeper with electron microscopy without any fractures inside. So the second, more challenging work was with worms, with adult worms that are quite challenging because with a standard cryopreservation they do not survive. So after the application of this ultrasound, you see after 70 seconds of 60 seconds of treatment, from nothing, from zero recovery rate, you get almost 100 recovery rate of this adult C. elegans. And of the same graph, but plotting against the warming rate by increasing the power of the ultrasound. And you can see here a couple of these worms finally growing and after surviving this treatment. And finally, the more this appeared only a few months ago that we want to go to a transplantable organ. In this case, it was a mouse heart. So we took the hearts and went to deep cryogenic, deep hypothermia, not cryogenic, -6 degrees. And then what we did was just to make the warming with the ultrasound and the warming in the regular way. So with the ultrasound is almost instantaneous. The regular warming is 5 degrees per minute. And this is the performance, 100 degrees, 100%. The recovery of the beating rate with the ultrasound, and practically nothing if you leave it on air for a few minutes. So this is also the internal structure that are perfect, are without any problem. And this is one of the hearts after recovery with the ultrasound beating regularly in our lab. So thank you. So the fields of this ultrasound technology are many: organ transplantation, ovarian tissue preservation, tumor banking, for example, can benefit from the discovery of drugs, the preservation of biodiversity. These are our facilities. This is the new machine that we are now, has 128 channels, not only 1 or 2 like in the past. So I can control each channel in an independent way, the frequency, the power, and so on. And this is the more important part. This is the human thing. These are the PhD students that are doing the real tough work, very enthusiastic on cryopreservation, and without them, this would have not been possible. And nothing else. I wanted to end this presentation with this fingerprint of the ultrasound after doing its job, fighting with a piece of ice after being in liquid nitrogen in 196 degrees below zero. And you see how nice was the warming making this hole. Thank you very much. It has been a pleasure. And
Thank you. We have 4 minutes for questions. Who would like to start?
Yeah, thanks. Really great proposal here. I think oftentimes the cryo field doesn't get enough. Everyone's saying we could be focusing more on rewarming. So I think it's cool that this project is really focused on that. I'm curious when it comes to scaling this, because I know in your list there you had everything from fertility preservation all the way to humans to cryonics. What do you just add more channels? Do you just make a bigger microwave? What does this look like?
The scalability is straightforward. I mean, once you know how they work, it is very easy to understand why it is totally scalable. The thing is that you have thousands of small transducers, and each transducer is controlled in the phase of the wave. So by doing interferometry, you decide how deep or how close to your transducer you put the energy. So it's a question of acoustic interferometry. So you decide where to put the energy. And how do you make that decision? Because you are monitoring by MRI with thermography by MRI in real time. So your MRI machine says to your HIFU machine where to put the concentration of the beams each time. So it can penetrate centimeters far. It goes in the deep brain. That's why you can treat Parkinson, tremor, or Alzheimer, or all these diseases, because you can burn inside any tumor in the brain. So it is totally penetrable and escalable, the technology.
For the rewarming, what volume are you rewarming whenever you're having?
Up to now, we are working. The maximum has been mouse hearts that are something like this, small, but in principle, it can be as big as you want. It does depend on the amount of power that you have in your pocket. With that machine, we have only 100 watts. But if you have something like 3-4 kilowatts, that is nothing, you can rewarm a very big sample.
That's at the end of your procedure. I mean, right at, let's say, an instantaneous moment, how much area or how much volume are you warming?
What I have had in the past has been the volume of a cryovial, that means 2 ml, for example,
2 ml.
but it was a proof of principle.
Okay,
It can be done with something like 1 liter easily.
Okay.
Something like any kidney, human kidney, or that's not a problem.
Maybe I'm not sure exactly how to ask this, but I'm assuming you used a cryoprotective agent when you cryopreserved the.
Yes, it is only for the rewarming. You have to put the cryoprotectant, and once you have put the cryoprotectant, you need to have a quick rewarming.
But the process is probably dependent upon the chemical that you're using as a cryoprotective agent.
No,
correct?
not at all, not at
No?
all.
Okay.
For any cryoprotectant, whatever you use, any cryoprotectant, you need to have the quickest rewarming as possible. That is a thumb rule.
Okay. And
So
so how much and how long?
How much what?
How much money do you need and how long will it take to get a result, do you think?
I did it with the money of my pocket, so this is, yes.
Okay.
These results are without any special funds. Only this machine, I got a grant for buying it. I am about to use it. This is the manufacturer. He manufactured for brain tumor burning inside he's in Burdeos. There are only 3 manufacturers. One is in China, the other is in Oregon, and this is in Burdeos. And this is our new machine that has 128 channels. So we can control with these 128 channels. We think things bigger than this small mouse heart. Things like rat organs or rabbit organs. This is the point. So this machine cost something like $250,000 to 250. This is the price of the machine, 250,000. But the result that I have shown in this slide was done by money of my pocket, practically.
And how long does it take to get a result?
You mean for the next product? I think if I have the help of someone here, probably very, very, very soon.
Next up, we have Ralph Spindler.
Good afternoon. My name is Ralph Spindler, and here I'm proposing a research project to investigate the effect of vitrification on rabbit memory.
The big button in the middle, yeah.
Okay. My background: I'm an electrical engineer and a cryobiologist, and I also worked in neuroscience. I immigrated from Germany to the US, driven by the passion of cryopreservation and cryonics. Currently, I'm conducting research in the field of brain cryopreservation at 21st Century Medicine, thanks to Dr. Greg Fay, who brought me on board. The overall goal of our studies is to find a reversible cryopreservation protocol that allows us for our survival. This protocol can greatly impact future technologies, such as organ banking, space travel, and allows us to benefit from future medical treatments like rejuvenation. A promising approach in this regard is an ice-free cryopreservation method, also known as vitrification. why are we performing research on rapid brain cryopreservation? On the left-hand side, you can see a worm, C. elegans, which has been successfully cryopreserved with retention of memory after vitrification. However, these worms do not have a brain. On the right-hand side, you can see a human brain. Even if we could cryopreserve human brains successfully under ideal conditions, rewarming and testing them for viability would be non-compliant with regulatory standards. Therefore, we are conducting research with rabbit brains. They are cost-effective, compliant with regulations, and their size is sufficient to prove that brain cryopreservation is reversible. Here I lay out the 3 pillars of brain cryopreservation research: ultrastructure, viability, and memory. 21st century medicine has been conducting research in ultrastructure over 20 years. My team and I are currently researching rabbit brain viability, and we have some substantial advances there. Dr. Greg Fay, he's sitting here, he will give a speech tomorrow about some exciting data in ultrastructure and viability, and we plan to publish the ultrastructure data soon. We aim to add memory as a 3rd pillar. Why? What's the point of bringing us back without our memories? We also like to use synergy effects to speed up cryopreservation research, meaning results from one pillar can greatly impact and positively impact the other pillars. So we cannot simply cryopreserve a whole rabbit, rewarm it, and measure memory directly because there are technological limitations, and also this is non-compliant with regulatory standards. But what we can do, we can measure memory indirectly by investigating key molecules which are associated with memory, such as proteins, receptors, neurotransmitters, which are critical for memory storage. How can we analyze them? We can use Western blotting or specific imaging modalities, such as mass spec imaging. Without key molecules, memory does not exist. Here I lay out the proposed project. On the left-hand side, you can see what we are doing at 21st century medicine on a regular basis. On the right-hand side is what we like to add to our project, and which we are seeking funding for. Starting on the left side, we use rabbits, isolate their heads, vitrify their brains, fix them chemically, and create hippocampal tissue slices. We also run controls. We like to get additional budget for an imaging method which allows us to measure key memory molecules for vitrified brain tissue samples and controls. Then we need to compare both sets of images, and we need to see if there are significant changes between the distributions of these key memory molecules. Therefore, we need a bioinformatician, an analysis software to do the data analysis to prove that vitrification preserves key memory molecules. In summary, how to find a reversible cryopreservation protocol for our survival fast, First, we need to publish the ultrastructure data. Then we need to continue with the viability investigations, and we need to start the memory project as soon as possible. Let's start now, since time matters. Thank you.
Hold on to that. All right. Great. Okay. Great job, Ralph. You're the only one who had about 10 seconds to spare, so we have a full 5 minutes for questions.
Hey, awesome. So let's assume you can prove that these molecules are still there, right? So that's probably required to prove that memory is preserved, right? But that's probably not sufficient, right? How much of the way there do you think this is? Let's say you have good ultrastructure preservation, and the molecules are still there. Like, and one more question. Having good ultrastructure preservation, does this also mean that these memory molecules are most likely there, and the other way around? Or are these totally independent variables?
Thank you for the question, because I think this is exactly what I mentioned before. Ultrastructure needs to be first. This is most important. And if we are, and Greg will talk about ultrastructure tomorrow, if can show that ultrastructure works, this is the prerequisite, basically. And then the memory is, I would say viability is the second step, and memory is the third step. So without these, the first and the second pillar, the third pillar would not make sense. I mean, however, it makes sense to start them interleaving, not do one pillar and then when it's finished, start the second one and then the third, because we can learn from each other. And that's what we realized when we worked, since we worked now on viability, and we realized, wow, there is something like SDS, we have to do some changes there on the ultrastructure side. And so that's really a big thing here. And going back to your other question, this was about if independent variables, or if
yes,
say
you have the molecules, right? Is that then, if you have ultrastructure and you have the molecules, would that then, is there like either an unknown unknown or a known unknown?
So what I can tell you to that question, from the ultrastructure side, we realized, for example, that some parts in the extracellular space are washed out. We named them, they are also known as white spaces. There are spaces with low density. We think it's caused by osmotic damage, or, no, sorry, I have to say it differently, osmotic effects. It's not a damage. And we think it is reversible. And a comparison with studies from medical neurosurgeons on humans, where the surgeons shrink the brains of humans with mannitol, for example, to get access to the brain. And we use the same concentrations in our rabbit brains, and we see also we see these white spaces, assuming that in the medical field these humans also have these white spaces, and they live afterwards, and they don't appear to have any effects. So yeah. But about these, if we find, for example, the controls and the vitrified samples have the same distribution and concentration of these key memory molecules, yes, then this would be a success.
You're working on it. You're working on another project. What is the other project you're working on at 21CM?
I'm working on the first two pillars, basically.
The first two pillars.
Yes.
Okay. How is that the new project going to impact your work on the other one?
I think it's very nicely adding on it. We are just going to finish the ultrastructure studies, and we don't forget them, of course, hopefully. But the second and the third pillars, they work then very well together. And then, for example, we would basically we need all the technology we as our 21st century established over the many years, decades. This works is basically a foundation for the memory study then. So it will, I think it will, like I said here, it will speed up cryopreservation research because we don't look back and have to change something in the first pillar.
I guess I was just asking, is it going to take away from your research doing the second pillar to do the third pillar already?
I don't think so. If we get at least one person who's doing the data analysis in-house, and if we outsource what we don't know all, like the technology, like mass spec imaging, we don't know, we just outsource it. And therefore we need the main portion of the budget.
Thank you. Okay, I have to give a quick shout out to our next researcher because we were actually supposed to have Roman Bauer here for the last pitch, but because of all the flight issues, he wasn't able to make it, unfortunately, and so Borys stepped in very last minute to prepare a presentation. So thank you very much, Borys Wrobel.
All right, so I'm not Roman Bauer. My name is Borys Wrobel, and I will be talking about preservation of large mammalian brains, and large mammals here include humans, with connectomic quality. So the methods that we used have already been mentioned by Max. Cryovitrification, that's the method that we use for preservation brains at connectomic quality. And large mammals here in this presentation would include pigs and dogs, apart from humans. All right, so physically, I'm based in the Netherlands. We created a new research organization called European Institute for Brain Research, which is a non-profit. And I work together with Nectom, which was originated by Robert McIntyre, now Aurelia Song. We work together in those two areas, the west coast of the US and the Netherlands, for the very important reasons that these two jurisdictions allow for either euthanasia or medical assistance in dying, which is very important for the method that we use. So the method of preservation that we use has to be a planned procedure, and perfusion has to be initiated extremely quickly in order to allow connectomic quality preservation of the brain with aldehydes. All right, so feel free to access the webpage of the European Institute for Brain Research if you're interested to learn a bit more about our team, including myself. My background is academic. I have a PhD in biology. I worked in computational neuroscience also, and now moved from academia to this field. And as you can see, we are launching now our human donation program. So in the Netherlands, what we will plan to do is to work as a non-profit, an organization to which humans will be donating their whole bodies, including the brain, for research. And that would allow us to validate the methods in humans. So previous research was done in pigs in the US, and we shown that our method can be actually used in the field in a manner that is compatible with post-mortem situation in humans after euthanasia. So in order to do that, we have a mobile unit, essentially an ambulance that has been adapted to deliver in the field preservation with aldehydes, which presents, as Ashwin mentioned during his presentation, some technical challenges that we solved. So there are several technical challenges there that are different from cryopreservation with vitrification mixtures, one of which the necessity of a very rigorous washout. There cannot be any blood cells present, or a very minimal number of blood cells, because that can embolize the brain. So that's one important thing. The other is safety. So we are having aldehydes there, which are toxic to humans. We have to ensure the proper ventilation and so on. This is not an issue for vitrification. Vitrification measures the safety of the team that delivers. So this vehicle now in the Netherlands has been equipped. It's fully equipped with a perfusion machine that I've finished constructing over the past several months. We have something like an ice bath situation here. It's a mobile operating room that we already used to test using cadavers, human cadavers, and we did obtain brains. You can see a picture of a fragment of a brain that we already obtained. This picture is like to show the interesting part. It's taken from the visual cortex, so you can see here a layer of cells that are receiving the information from the thalamus. That's a very characteristic feature of a part of the brain that receives the input, the visual input. All right, so our research with pigs have been previously funded by BLRS. I would like to acknowledge the financial support. And we used pigs in order to validate the methods and to test the perfusion machines that we built. And pig number 8 here is a perfect connectomic level preservation. In some of the pigs that we researched, if the window from what we call the perfusability window was too long, essentially longer than 20 minutes, then sometimes it's the preservation is not perfect. You can see some areas in which there's like a washout of structure. So we consider that an imperfect preservation. But if the perfusion is initiated very shortly after cardiac arrest, we can achieve a whole brain completely preserved connectomic quality, traceable at the level of axons and individual synapses throughout the whole brain. So we don't have to pick and choose regions. All right, so what we are working on, what we looking for funding on. So one is more like RD work in which we're optimizing protocols, miniaturizing equipment, reducing dead volume. That seems trivial, but it's technically important. So if we want to deliver in the field, we need to limit the number of solutions, the total volume of waste also used. That requires a recirculation of the cryoprotectant of the solutions, and then the dead volume becomes an issue. You don't want to spend time introducing cryoprotectants. You want to concentrate introducing the chemicals as much on the body and not on the volume of liquids that are simply sloshing around in your equipment. The other part of the research that we're working on is expanding this perfusability window. So as I mentioned, in pigs, we have also in our rat work, essentially 20 minutes is a cutoff. And it's to be seen whether in humans it's a similar situation or whether we can extend this window in which the brains can be preserved completely. Aldehydes open a possibility of high temperature storage in liquid. So it's not necessary to store with aldehyde at minus 130. It could be still ideal to preserve it around the glass transition temperature, but it's not strictly necessary. And also temperature excursions are in principle possible. So if your preservation fails for some reason, liquid nitrogen evaporates or whatever, then the brains and bodies preserved in this method would be still stable. So it's less of an issue. And we also want to build infrastructure for brain banking and preparing anatomical specimens, making that available to other researchers who are using those methods. And finally, we are ready to launch biopreservation as a service, and for that we need actual investors, not so much donors, but talk to me if you would actually be interested in investing and making it like an available technique for clients to be able to do it. And we're working with Brain Preservation Foundation as an outside organization that will be controlling the quality of what we deliver. So that's why I put those on the page. And feel free to use those manners of contact me if you would like to ask me some more questions that you're unable to ask here, or to follow up on the presentation. Thanks.
Thank you. We have 3 minutes for questions.
So you said you want to expand the window of opportunity, the perfusability window, right? Like what are you planning to do there?
So the easiest one actually is to cool, surprisingly, right? So if you cool, we've demonstrated I'm not exactly necessarily saying that that's the easiest to deliver in practice, but that's one way of doing it. So in the rats, at least, in my experiments, if you would cool the rats and ventilate, that increases the window of perfusability. Now in practice, as you know, like if you have access to the body and you can cool, you can go ahead and don't bother with it. You can already start opening the chest and introducing the chemicals. So I'm not necessarily saying that that's one way that's gonna make an impact in practice. Because in practice, the issue is, as you well know, in practical approach to and delivering it in the field, is to accessing the body quickly enough. That's the main bottleneck for successful preservation. So nobody wants to ask me how much I want.
No, that's why I just took the phone, the microphone. Like how much money are you raising?
All right, so it depends on what. Like, do we have this? I have it actually. Okay, so
for biostasis as a service.
So expanding the postmortem window and high temperature storage, this is what we consider a low funding type of level. That's probably something to the order of 60K. Would allow us to do some progress there. For high temperature storage and ice detection. So this is like expanding a postmortem window and brain banking ideas that would require something in the order of $500,000 per year for a year project to make that deliver, and to launch our to fund the funding for the company that we plan to launch. So an investment thing, we are looking for fundraising at the level of about 1 million, 1.2 million.
Thank you so much.